Hello and welcome back to the Biochemistry for Health Sciences channel. In the last video, we discussed how insulin works. In this video, we shall continue with this discussion. So in the previous video, we discussed how insulin increases glucose uptake and various aspects of carbohydrate metabolism, including glycogen synthesis, glycogen breakdown, as well as gluconeogenesis. So in today's video, we'll see how insulin affects lipogenesis, which is the increase of triglyceride synthesis, lipolysis, which is triglyceride breakdown, as well as protein synthesis. As discussed in the previous video, we'll see how insulin uses a variety of strategies to affect metabolism of lipids as well as proteins. And this included strategies such as sequestering enzymes, allosteric regulation, covalent modification, and affecting transcription and thereby changing the concentration of key metabolic enzymes as well as transport proteins. So let's begin by discussing number 5 here, the effects of insulin, and that is how does insulin increase lipid synthesis, triglyceride synthesis especially. And this is important process that happens in the liver where a lot of lipids are synthesized as well as the adipocytes. So let's look at how insulin first increases lipid synthesis, and we will begin with how it does this in the liver. Now in the liver, insulin will increase the transcription of this protein here called SREB1C, which stands for steroid regulatory element binding protein. And the increase in this protein will lead to the increased transcription of several enzymes that are involved in lipid synthesis. For example, there is increased transcription of the enzyme acetyl-CoA carboxylase, or ACC, and as we discussed in a previous video on fatty acid synthesis as well as triglyceride synthesis, this enzyme plays a very important role in the synthesis of fatty acids. So this enzyme concentration will increase, thereby increasing fatty acid synthesis. Besides this enzyme, then the enzyme FAS, fatty acid synthase, is also increased. And this is another enzyme that's involved in fatty acid synthesis. And once again, as the concentration of this enzyme increases, fatty acid synthesis increases. There's also increased transcription of the enzyme glycerol 3-phosphate acyl transferase, or GPAT. And this enzyme is responsible for incorporating the palmitic acid into glycerol, forming triglycerides. So as the concentration of this enzyme increases, triglyceride synthesis in the liver increases. There is also increased synthesis or transcription of the enzyme glucokinase. This is the enzyme that's responsible for converting glucose into glucose 6-phosphate, which eventually becomes a pyruvate, enters the mitochondria, becomes acetyl-CoA, and that acetyl-CoA will be transported out into the cytoplasm where fatty acid synthesis occurs, and therefore there will be increased acetyl-CoA concentration in the cytoplasm. And this acetyl-CoA, as we saw in a previous video, is the building block for synthesis of fatty acids. So all of this is going to increase fatty acid synthesis as well as triglyceride synthesis in the liver cells. So besides affecting transcription of various enzymes, insulin also causes the activation or inhibition of enzymes by covalent modification. For example, this enzyme acetyl-CoA carboxylase or ACC exists in the phosphorylated form, and this phosphorylated form is the inactive form, and it also exists in the non-phosphorylated form, which is the active form. So insulin will inhibit this enzyme AMPK, and AMPK therefore will not phosphorylate the ACC. So most of this ACC will be present in the non-phosphorylated active form, and since this is the active form, fatty acid synthesis will increase, and that will lead to eventually to increase triglyceride synthesis. Now this enzyme ACLY or ATP citrate lyase, which will break down the citric acid in the cytoplasm to give you the building block for fatty acid synthesis, acetyl-CoA, this enzyme also exists in the phosphorylated form as well as the unphosphorylated form. So the phosphorylated form of this enzyme is the active form. Now insulin will phosphorate, insulin signal transduction will lead to phosphorylation of this enzyme. It will lead to more and more phosphorylated ACLY, which is the active form. And this activity increases, breaks down the citrate to give you the building block, acetyl-CoA, which will be used to make the fatty acid. So that will lead to increased fatty acid synthesis. Now let's see how insulin increases triglyceride synthesis or lipogenesis in the adipocyte. Recent research shows that insulin increases lipogenesis in the adipocytes mainly by increasing fatty acid esterification. That is basically taking fatty acid, combining it with glycerol to form triglycerides. So fatty acid esterification. So what insulin seems to be doing is that it increases the activity of this enzyme lipoprotein lipase. And lipoprotein lipase, as you know, which we discussed in a previous video, is found in the endothelium of blood vessels in tissues. So for example, in adipose tissue endothelium, this enzyme is activated, and that will lead to the breakdown of the triglycerides in chylomicrons. Remember, after a meal, we have a lot of chylomicrons coming from the intestine. And these triglycerides in the chylomicrons will be broken down to fatty acids and glycerol. So another thing insulin does is that it increases the transport of these fatty acid transport proteins or FATPs from the perinuclear region to the plasma membrane. So FATP transport is increased by insulin to the plasma membrane, and once these FATPs are embedded in the plasma membrane, these fatty acids can be transported or imported into the cytoplasm. So now with the fatty acids available and glycerol coming from glucose, we can esterify these fatty acids, combine it with glycerol to form triglycerides, thereby increasing lipogenesis. So this is the major strategy that insulin uses. Increases lipogenesis in the adipocytes mainly by fatty acid esterification. There is another minor process, a minor way by which insulin can increase lipogenesis, and that is de novo lipogenesis. So basically what insulin does here is insulin increases the synthesis or the transcription of several transcription factors, especially SREB1C and the transcription factor PPAR gamma, which is peroxisome proliferator activated receptor gamma. And these transcription factors will then lead to the increased transcription and synthesis of key enzymes involved in triglycerides, fatty acids, as well as triglyceride synthesis. And these enzymes include enzymes like acetyl-CoA carboxylase, fatty acid synthase, glycerol 3-phosphate acyl transferase, as well as hexokinase 2, which converts glucose to glucose 6-phosphate, providing the glycerol necessary for esterification of the fatty acids to triglycerides. So this is another way by which insulin can increase triglyceride synthesis in the adipocyte. Although this is a minor way of doing it, the major way of doing it seems to be fatty acid esterification. Okay, so now let's look at how insulin decreases triglyceride breakdown, lipolysis. Now remember, most of the triglycerides are stored in adipose tissue, okay, not in the liver. So we will focus on how insulin breaks down triglycerides in the adipose tissue. Now, in order to understand how insulin suppresses lipolysis in the adipose tissue, we must first understand how this enzyme, protein kinase A, actually stimulates lipolysis. So once we understand how pKa stimulates triglyceride breakdown or lipolysis, then we'll be in a better situation to understand how insulin suppresses lipolysis. So in the presence of cyclic AMP, and we'll talk about this in a future video, this enzyme protein kinase A, pKa, becomes active. And pKa then goes on to do a few very important things. So one of the effects of pKa is that it will lead to the phosphorylation of this enzyme hormone-sensitive lipase, HSL. And this is the enzyme found in the fat droplet in the adipocyte that will break down the diacylglycerol into fatty acids and glycerol, thereby increasing triglyceride breakdown or lipolysis. So when HSL is phosphorylated, the HSL will move from the cytoplasm and will be transported to the lipid droplet. So it will now be transported to the lipid droplet, and once it's on the lipid droplet, it will start breaking down diacylglycerol, which came from triglycerides, into fatty acids and glycerol. Now, another major effect of pKa is that pKa phosphorylates this protein called perilipin 1 or PLIN1. Now, PLIN1 plays a very important role in lipolysis, and let's look at that. Now, one of the important functions of PLIN1 is that the phosphorylation of PLIN1 is required for this enzyme here to be active, this hormone-sensitive lipase or HSL. So PLI1 phosphorylation is required for activation of HSL. There is another enzyme, and this enzyme is adipose triglyceride lipase or ATGL. Now this enzyme, together with its cofactor CG158, in the presence of the cofactor, this enzyme will break down triglycerides into diglycerides, diacylglycerol. And this diacylglycerol or diglycerides will then be broken down by HSL into fatty acids and glycerol, thereby increasing lipolysis. Now, normally this cofactor CG158 is bound to PLIN1. So this cofactor is not available to this enzyme. However, when PLIN1 becomes phosphorylated, so when PLIN1 is phosphorylated, this binding is weak and the CG158 is free. It is removed from the PLIN1, and now this CG158 is able to bind and activate this enzyme ATGL. So now the cofactor will be bound to ATGL, and in the presence of this cofactor, the ATGL activity increases tremendously, almost 20-fold. So that is another way by which phosphorylating PLIN1 leads to increased lipolysis. Now, triglyceride hydrolysis happens on lipid particles. So the surface area of these particles is important. Okay, so the more smaller droplets we have, the larger the surface area. So one of the effects of PLIN1 phosphorylation is that it increases the lipid droplet surface area to volume because PLIN1 phosphorylation stimulates the budding of lipid microvesicles. So basically this big lipid droplet results in budding, forming smaller and smaller droplets. And since many of these enzymes, HSL and ATGL, they work on the surface of these droplets. So by having a larger surface area, this lipolysis will increase. So that's another way by which phosphorylation of PLIN1, perilipin-1, by protein kinase A leads to increased lipolysis. Okay, so now that we know how protein kinase A leads to increased lipolysis, increased breakdown, now we can see how insulin will suppress lipolysis. So we'll be in a much better situation now to understand how insulin suppresses lipolysis. Okay, so one effect of insulin is that it stimulates this phosphodiesterase PDE3B. And since cyclic AMP is a powerful allosteric activator of protein kinase A, by breaking cyclic AMP into AMP, this activation is not going to happen. Protein kinase A is not going to be functional, and therefore lipolysis will be dramatically reduced or even stopped. Another effect of insulin is that insulin stimulates the enzyme HSL phosphatase. So remember, HSL, if it exists in the phosphorylated form, the phosphorylated form is the active form. This is the active form. But what insulin does, it activates the enzyme HSL phosphatase, which removes this phosphate and makes HSL in the inactive dephosphated form. So if HSL doesn't work, diacylglycerol or diglycerides will not be broken down and lipolysis will be suppressed. And another powerful way by which insulin suppresses lipolysis is that it dephosphorylates perilipin. So perilipin is only functional when it is phosphorylated. So by increasing the dephosphorylation of perilipin, all of these effects will not happen, and thereby lipolysis will be dramatically suppressed in adipocytes. Okay, so that's how insulin affects lipid metabolism. And finally, let's look at another effect of insulin, and that is how it increases protein synthesis. And most proteins are synthesized in the liver as well as the skeletal muscle, so we'll focus on these two tissues. First, let's look at how insulin increases protein synthesis in the liver. Now, the binding of insulin to its receptor leads to the activation of PKB, and one of the effects of this activated PKB is the activation of this network called mTOR, or mammalian target of rapamycin, mTOR. This is a very huge, very large protein kinase that is found as complexes. These two complexes especially, mTORC1, which is the most studied complex, as well as mTORC2. Now, mTOR, this mammalian target of rapamycin, is a master regulator of several processes in the cell, including cell growth, as well as various metabolic pathways, as well as autophagy. And we discussed mTOR in a previous video 5.28. So insulin basically will activate, especially this mTORC1, which is the most studied. And the activated mTORC1 then will go on and phosphorylate several components of the protein synthesis, or the translational machinery. And this includes, this will result basically in increased ribosome synthesis and assembly. So the more ribosomes are synthesized and assembled, the more proteins will be made. It will lead to increased mRNA synthesis, which means more proteins will be made. And it also increases the synthesis of several factors that are very important in protein synthesis or translation. And we discussed early in an earlier video. And these factors include initiation factors as well as elongation factors. So all of these effects will lead to increased protein synthesis in the liver. Now, how does insulin increase protein synthesis in the skeletal muscles? Recent research shows that the major effect of insulin on skeletal muscles might be that the insulin affects the blood vessel endothelial cells as well as the smooth muscle cells, and that leads to vasodilation. The blood vessels increase in size, vasodilation. That means more blood is going to flow to the muscles, and that means there's going to be more amino acids in the blood vessels of the muscles, which means the amino acids will be transported or will enter the muscle cells, and they eventually lead to protein synthesis. So this seems to be one of the major effects, although there is a minor effect that also has been reported, and that is through the mTOR pathway, which will lead to increased protein synthesis. So to summarize the effects of insulin on the various parts of the body. So after a meal, when glucose is being absorbed, your blood glucose concentration is high. That will trigger the release of insulin, and insulin will cause a variety of effects. It will increase glucose uptake in the liver. It will increase glucose uptake in the muscle. It will increase glucose uptake in the adipocytes. Insulin will also increase glycogen synthesis at the same time, decrease glycogen breakdown in the liver as well as in the muscle. Insulin will also stop gluconeogenesis in the liver, and it will increase lipid synthesis in the liver as well as in the adipocytes. In adipocytes, it will also prevent lipid breakdown. So while lipid synthesis is being increased, lipolysis or lipid breakdown will decrease in the adipocytes where most of your fat is stored. And finally, as far as protein synthesis is concerned, insulin will increase protein synthesis, especially in the liver as well as the skeletal muscles. So this is a summary of how insulin controls metabolism after a meal. So until the next video, thank you once again for watching and supporting this channel. Have a great day, and hopefully we'll see you soon.